How do we distinguish living organisms from non-living organisms? There are several characteristics used for this differentiation, but one of the most important is related to reproduction of organisms. Now this reproduction is based on cell cycle parameter, which is basically division of cell after some certain interval of time. What is this cell cycle and how it is important for reproduction? That is a major aim of this module. But again, this cell cycle, it is a very basic topic in biochemistry and some other topics related to molecular biology. So we will not go into details of cell cycle alone. Our focus will be how cell cycle operates on drug discovery mechanism. For that purpose, we should know that cell cycle has a direct effect when we are dealing with cancerous or normal cells. इस मॉड्यूल में हम ये देखेंगे कि what is cell cycle, what are its different types, and how it has effects on cancerous cells. So basically, what is a cell cycle? By definition, we can say that cell cycle is a series of steps that takes place for cell division. Now this cell division, it can be of two types. What are those types? That can be basically asexual reproduction or sexual reproduction. But before that, whenever we talk about cell cycle, there are several different steps, which are interphase, in which cell performs its normal function. Then we have mitosis. Mitosis is the phase in which the cell divides into two different cells. And finally, we have cytokinesis, which is basically division of cytoplasm inside the cell. Moving on about cell types, cell cycle types. So before I say what are the cell cycle types, I will define two things, that cells are of two organisms, which are eukaryotes and prokaryotes. So for prokaryotes, the common technology used for cell cycle is binary fission. Binary fission, basically, as name suggests, it is just breakage of cell into two cells with the same structure. On the other hand, in prokaryotes, there are several other things happening during the cell cycle phase. For example, in mitosis, which is, I said there are two types of cell cycle. One type is mitosis, which is basically asexual reproduction, in which two cells are produced which have the same genetic structure as their parent cell. On the other hand, the second type is meiosis. What happens in meiosis? It is basically sexual reproduction, which yields genetically different cells with half the genetic information from one parent and half the information from the other parent. So they occur in specific reproductive cells such as eggs and sperms or spores. So what is the effect of cell cycle and how it is related to the disease? So whenever we talk about disease, what is a disease basically? Disease is basically a bad version of a cell, where there is not a good productive functioning happens for a cell. So definition wise, we can say that cancer is a genetic disease with uncontrollable growth of the given cell. कि अगर हमारे पास एक सेल है और उसकी जो ग्रोथ है वो नॉर्मल नहीं है, मे बी दैट growth has increased or decreased, but that is not normal, that we say that it will result in a cancerous cell. So for that purpose, there is a cancer genome project. Basically, this cancer genome project is somehow similar to cancer genome atlas. Cancer genome atlas basically is a dictionary which contains definitions of different cancers, their types and their behavior. And cancer genome project, it basically is parallel to human genome project, but the idea is that out of all the genes which are related to humans, find out those genes which can cause cancers in human beings. So in cancer genome project, until now we have 30 tumor suppressors and 100 dominant oncogenes. Now what are these dominant tumor suppressor and what are these dominant oncogenes? So suppressor, as the name suggests, these are the genes which help the cell to come out of cancer. कि कैंसर से कैसे बचा जा सकता है? Those are tumor suppressors. They are also called anti-oncogenes. On the other hand, dominant oncogenes are those genes which have the potential to grow in a cancerous cell. Here I have given an example of cell cycle in proliferative diseases. On the left side, this figure represents a normal cell. So here we have a cell surface marker which tells outer boundary of the cell or starting of the cell, and here we have nucleus in which we have different chromosomes, and it is a normal cell. Now what happens on the right hand side? This one basically is a cancerous cell in which there is a cancer. Now what happens is that we have different abnormal conditions which cause distorted protein activities by a faulty gene expression and alter intercellular signals such as shown here. So if signals between different cells they change, they affect this information as shown by this cancerous part in this particular example. So what happens is once this cell converts into a cancerous cell, then it moves toward a state which is called apoptosis. So apoptosis means that death of the cell. So why this death occurs? Because of somatic mutations followed by genomic instability due to deletions, translocations and amplifications. Now what is the status of current cancer therapies? So current cancer therapies, basically they are very good while controlling the diseases and affecting or curing different diseases, but they have side effects. So what are those side effects? Such as there is no discrimination between normal and cancerous cells. So okay, if we take a medicine for some disease, it will remove the disease from the body, but what will happen? It might produce some cancerous cells in its cell cycle stage. So what happens? They act on cell cycle which causes perturbation of cyclic events. This perturbation, what happens? That oncologists basically those persons who are biologists who are working to remedy different cancers from cells, they suggest that there should be cell cycling and regulatory pathways which consist of different proteins to control these cancerous cells. So finally, there are therapies for selecting and validation of cancer cell cycle targets. So if we have cell cycle targets and identification techniques, then they can be used for identification or drug discovery.